Okay, in this video, we're going to be summarizing what happens in a muscle using all the anatomical terms that you learned in the longer lecture videos. First off, we have the sarcolemma of a muscle cell. That's just the plasma membrane, but remember the name changes in muscle. Everything has to be called something else because muscle is just special. Muscle cells are fairly bulky, so in certain places, the sarcolemma is actually going to dive into the cell. Those places where that happens, they are called T-Tubules. And I'm even going to label that poorly, of course. So there's a T-tubule. Sitting next to the T-tubule, and I'm going to change colors here, we have Sarcoplasmic Reticulum, which is the same thing as smooth endoplasmic reticulum, but here in muscle, it has a very important function, and that is it stores calcium ions. Calcium ions are going to be our trigger for muscle contraction. If calcium is present in the cytoplasm, muscle wants to contract. So ordinarily, we keep the calcium locked up in the SR until we are ready for muscle to contract. Now, in addition to all of that anatomy, we also have out here all of these nice little sarcomeres. Now, I'm only going to abbreviate my drawing here, and it's going to be, unfortunately, a very messy drawing because I am not an artist. We have thin filaments, and then we have thick filaments. The thick filaments have heads. The head wants to be able to grab the thin filament and then pull that thin filament closer to the midline of that sarcomere. But ordinarily, there is a gate that blocks the myosin head. That gate is called tropomyosin. The gatekeeper is troponin, and what tells the gatekeeper that it's time to unlock the gate is calcium ions. So here is what essentially is going to happen, and I'm going to switch colors one more time. So, a motor neuron is going to release a chemical called acetylcholine. It's the first neurotransmitter we are going to mention in here. Acetylcholine binds to receptors on the sarcolemma and starts a signal. That signal is going to travel down the sarcolemma, down the T-tubules, and it's going to tell the SR to dump the calcium. Calcium is going to flow out into the cytoplasm, and then it's going to bind to troponin on the thin filaments. The troponin is going to pull tropomyosin off the binding sites. Once that happens, the myosin head, go away, can grab onto the actin and then pull it towards the midline of that sarcomere, and that is muscle contraction. When it's time for the muscle contraction to be over, this signal stops. Calcium gets pumped back in off the troponin. That moves the gate tropomyosin back over the binding site. So now myosin can't grab again. The muscle relaxes and it goes back to its original resting length. So that is what happens during muscle contraction. I also wanted to show you, there is a nice clean picture of everything that I just depicted. I don't happen to know what page it's on in your textbook, but look for this picture. This is our motor neuron releasing acetylcholine, which are those little blue dots. It starts the signal, which travels down the sarcolemma, down the T-tubules to the SR. The SR dumps the calcium. The calcium binds to troponin. Troponin pulls tropomyosin off the binding site so that now myosin can grab actin and then pull. When we're done, the calcium gets pumped back up. Myosin lets go. We go back to our original resting length. I hope that helps you understand how muscle is going to contract.